Alright, so in the last lecture I mentioned relatively briefly the work of Till and Mcculoch and how that kind of gave rise to an understanding of hematopoietic stem cells and indeed their research on radiation and how that affects stem cells. Today I'm going to give you a little bit more background, a bit of a more deeper understanding, a bit of a deeper understanding of their research, what precisely they did and what precisely the implications of that were. So, as I discussed in the first lecture of this series, feel free to go back and see that if you want to. They took a mouse number one, took a mouse and they took its bone marrow. Right, now I'm going to do the 1961 experiment and then we'll move on to the 1963 and the 1968. So in 1961, they took that bone marrow, they put it into a new mouse which had been irradiated and its stem cells were dead. So like we said in the first lecture, this second mouse was able to act as a kind of living petri dish for the stem cells. And what we did, right, was we implanted this bone marrow into the irradiated mouse. Try and draw a nucleohazard sign here, although it's not necessarily correct. Right, they put it into that mouse, and then after 10 days, this is important, 10 days, they sacrificed the mouse and extracted its spleen, right? Now you take out the mouse's spleen, and you see all these nodules on it, right? Nodules on the spleen, you know, kind of like this sort of deal, right? And those nodules are cell colonies. Those nodules are groups of cells that have come from this transplant, probably, hypothesized, and grown on the spleen. Now that has a whole bunch of possible implications, but there's no hard evidence for a number of things that we need to verify from this particular experiment. So, now we need to see, and this is important, right? So, those nodules were shown to have a lot of the myeloid lineage, but not a lot of the lymphoid lineage. And HSCs should be able to give rise to both fully. So that's a bit confusing. Also, there was no evidence that these nodules came from one individual cell. So what that means is, what we could have had is a large number of cells migrating from the bone marrow and going into the spleen. And those large number of cells could have been myeloid progenitors and not hematopoietic stem cells. Now that's important because it's desirable for us to prove whether this was actually stem cell activity, right? Because we want to know whether or not giving this transplant was actually able to transplant the hematopoietic stem cell. And indeed, that stem cell retained the ability to proliferate and give rise to its whole lineage of cells. At the moment, we know that something is giving rise to a whole bunch of cells, but we don't know whether that is a single stem cell able to give rise to its whole lineage, or whether indeed it was just a whole few myeloid progenitor cells. Now, the difference between progenitor cells and stem cells is that progenitor cells cannot give rise to themselves. After about 3 generations, they die off. They can give rise to other cells, but they die off around after 3 generations-ish. Right? So how do we verify that the colonies that we have in the spleen are in fact stem cells. Okay, so what we do is, we get our first mouse, which I shouldn't have erased. Get our first mouse, alright. And we take our sample, and we see in the sample our little chromosomes, right? We also have our second mouse, which has been irradiated. I'm terrible at drawing mice. Right, and this mouse has already been irradiated, right? So he has no HSCs. No HSCs. Now what we've done is we take this first sample from the first mouse and we irradiate it. Now why would we irradiate those cells when we've already irradiated these cells to kill them? Well, you irradiate them less. You irradiate them less intensely so that you induce some genetic mutations in these chromosomes. The idea being that you can visibly see the variations in the chromosomes when you radiate them with a moderate dose. Now that allows us to tell, statistically it's very unlikely that two cells there are going to have the same exact mutations, right? And so when we get to when we take this sample and implant it into this guy, right? You've got a whole bunch of cells, all of which have slightly different mutations from the radiation. Now what that means is, if we indeed take our spleen, you see that? If we get our spleen and these nodules, if those spleen nodules each have a genetic mutation that is the same in the whole nodule, we know that that nodule was given rise to by one cell. Right? How do we know it was given rise to by one cell? Because statistically, it's very unlikely that multiple cells are going to have the same mutation, and therefore, this whole nodule had to come from one cell. One cell. Right? And that's exactly what we found. We found that each nodule overwhelmingly had the same mutation, which means that each nodule was given rise to by a single cell. Right? Now that means that you're having some cell which is able to proliferate. But at this point, we didn't know if that was an HSC or indeed it was a kind of myeloid stem cell, right? Because we were only getting the myeloid lineage. So we need to be able to tell, we need to figure out whether we're also able to get the lymphoid lineage. And that was done in a 1968 experiment. So what did we do in 1968? Well, we determined that progenitors will die in vivo culture after 4 weeks, right? I should correct the previous thing I said there. We didn't know whether it was an HSC stem cell or a progenitor cell of the myeloid range, right? So because we only had that one lineage from myeloid, there are no myeloid stem cells. We just didn't know whether it was a myeloid progenitor cell or the HSC stem cell, right? So, progenitor cells progenitor cells die after 4 weeks in culture. That's important, right? Why is that important? Because it allows us to weed out progenitor cells. Remember, we know that those colonies in the pancreas of the second mouse come from one cell because they have the same mutation. We don't know if those cells were progenitor cells or stem cells. Now here, we have a way of dividing the stem cells from the progenitor cells. So this is what we do. We get our mouse. Really need to get better at drawing mice, don't I? Right, we have our mouse. We take a sample of its bone marrow. We put that bone marrow sample into culture, right? Into culture for 4 weeks. Actually, we put it in culture for 5 to 7 weeks, but it needs to be more than 4 weeks, right? So this process of putting it in culture shows us for certain that we don't have any progenitor cells going into the second mouse, right? Now we do the same thing as before. Well, we don't need to irradiate them, but we can, whatever. And we take this colony, which has survived for more than 4 weeks, and we put it into our favourite irradiated mouse. Right, so it goes into the irradiated mouse, right? And then, if we get points on the if we get colonies on the spleen, if we get colonies on the spleen, we know from the 1963 test that they had to come from one cell, and we know from the 1968 test, this one, that they came from stem cells. Therefore, they came from individual, multipotent stem cells, which are able to give rise to their whole lineage. In a further test, we verify that they're actually able to give both myeloid and lymphoid, but in this test, we just know that they're actually coming from stem cells. Right, so come up here. Mouse lives. Right, and then we therefore have stem cells. Therefore, it's a shit therefore, stem cells. Right, therefore stem cells. And that is the 1968 experiment, and that is how we verified hematopoietic stem cells in the first instance. Now these suite of experiments, assays, are really, really popular and have been popular for 60 years. It's kind of the way that you verify multipotency stem cells in the blood until this day. Cool, thanks guys.